In this section, we're going to be discussing some general principles regarding amino acid metabolism. Now, just to remind you, amino acids have the following structure. They have a central carbon, they have a carboxylic acid attached on one end, they have an amino group attached on the other end, and then they're going to have some side chain, some R group, which determines the structure and function of those amino acids. Now, where do amino acids come from? Well, a lot of them we get from our diet. Dietary amino acids are usually in the form of protein, either in animals or in plants, and those proteins have to be digested to individual amino acids by peptidases in order to be absorbed by our intestinal epithelium. So the peptidases are synthesized in the pancreas and in the chief cells of the stomach as zymogens, which is a word we use to describe inactive precursors of enzymes. One of these zymogens, pepsinogen, is produced by the chief cells of the stomach, and it's activated to pepsin by the acid environment of the stomach. The other zymogens are activated in the intestine. There's a protein called enterokinase, which is embedded in the cell membranes of the intestinal epithelium. Enterokinase cleaves trypsinogen, which is a pancreatic peptidase, to trypsin, which in the process activating it. And then trypsin is able to activate other zymogens. It's able to activate other trypsinogen molecules to trypsin, chymotrypsinogen to chymotrypsin, proelastase to elastase, and also the amino and carboxypeptidases as well. Now, trypsin, chymotrypsin, and elastase are called endopeptidases because they cleave within the peptide chain itself, whereas the amino and carboxypeptidases chew off amino acids from either end, from the carboxy or amino terminal end of the proteins. Amino acids are transported into the intestinal absorptive cells by a sodium symporter. And this goes against the concentration gradient and then down into the bloodstream. The way that this works is that we create a sodium gradient using the sodium-potassium ATPase. And this pumps sodium out of the intestinal mucosal cells, creating a gradient for sodium. And so sodium then moves down its concentration gradient into the intestinal mucosal cell and carries amino acids with it via the specific transporter. Now, what are the requirements for amino acids in our diet? Remember that protein synthesis requires 20 amino acids. Of these, our cells can synthesize 10 from other biosynthetic precursors. The remaining 10 amino acids are termed essential amino acids because they have to be obtained from the diet. The essential amino acids are phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. There's a little mnemonic that we can use to remember this, Private Tim Hall. If you can do those in order, then you can remember which of the amino acids are essential. Now, we have a star next to arginine, an asterisk next to arginine, indicating that arginine is not always an essential amino acid. Our bodies can synthesize arginine to some extent, but in childhood and periods of growth and so forth, the ability of the cells to make arginine does not meet the demand. And so under those circumstances, arginine has to be obtained from the diet. In addition to incorporation into proteins, amino acids can be used as precursors for nitrogenous bases of nucleic acids. They're used for neurotransmitters. They're used in the synthesis of porphyrins, which we'll talk about later in heme synthesis. They're used as nitrogenous head groups of phospholipids. Excess amino acids from the diet can be degraded, and the carbon skeletons can be converted to fuel via the TCA cycle and gluconeogenesis, as we'll talk about later. The excess nitrogen is excreted as urea. Now, dietary amino acid intake has to be adequate to fulfill all of these needs. And with this comes this concept of nitrogen balance. In order to be healthy, the nitrogen ingested has to equal the nitrogen excreted. So normal, healthy adults have a nitrogen balance of 0. That is, all of the nitrogen that we take out, for all the nitrogen that we take in our diet, an equal amount of nitrogen is excreted as urea in the urine. Now, if there's a positive nitrogen balance, that is, if you're taking in more nitrogen than you're excreting, that usually means that it's an active period of growth where you're adding protein mass to your body. In disease or malnutrition, where amino acids are being consumed for energy without being replaced in the diet, the nitrogen excreted is going to be greater than the nitrogen ingested, and that's a negative nitrogen balance, and that indicates a malnutrition state. Now, the first step in degradation of amino acids is that you have to remove the amino group, and we call this process deamination. And this occurs in two steps. The first thing that happens is you get transfer of the amino group from an amino acid to an alpha-ketoglutarate to form glutamate. And this reaction is called transamination and requires vitamin B6 as a cofactor. So here we have an example of aspartate donating its amino group to alpha-ketoglutarate to form glutamate and oxaloacetate. The amino group from the resulting glutamate can be removed by an enzyme called glutamate dehydrogenase, and this results in the formation of alpha-ketoglutarate again with an ammonium ion. This process works for other amino acids as well. In a two-step process, for example, we're able to take alanine, for example, transfer the amino group to alpha-ketoglutarate to form glutamate, and the alanine in the process is converted to pyruvate, which can be used in gluconeogenesis or in the TCA cycle as energy. The glutamate then is converted via glutamate dehydrogenase back to alpha-ketoglutarate, so that is recycled, and in the process we produce an ammonium ion. So the net reaction then is a molecule of L-alanine plus NAD plus generates a molecule of pyruvate plus NADH plus ammonium ion. And that's because the alpha-ketoglutarate is recycled in this process. So the net reaction of this is simply deamination of the amino acid. Now, some amino acids, when they're deaminated, are converted to intermediates of the citric acid cycle. We just showed you two examples of that. And thus, these are termed gluconeogenic because they can serve as precursors for gluconeogenesis. Others are converted to precursors of acetoacetate or beta-hydroxybutyrate, and thus they are called ketogenic. That is, they produce ketone bodies. Some amino acids are both gluconeogenic and ketogenic. Now, there's a very complicated chart that you see in the next slide, which shows the different ways that all of the amino acids can be degraded. It's not important for you to memorize this other than to appreciate that some of these form gluconeogenic precursors and some of them form ketone body precursors. The glucogenic amino acids are alanine, arginine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, histidine, isoleucine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. Well, you may say, well, how am I going to remember that list? Well, the easy way to remember it is there are only two amino acids that are purely ketogenic, that can't contribute to gluconeogenesis at all, and those are leucine and lysine, the two L's. Okay? Everything else can contribute to gluconeogenesis in some way. Now, there are 4 amino acids that are both ketogenic and glucogenic, and those can be remembered by the mnemonic PIT: phenylalanine, isoleucine, tryptophan, and tyrosine. Now, these processes of amino acid degradation are especially important during starvation because in those conditions, proteins can be used as fuel for oxidation. They can be used to form ketone bodies to provide fuel to the brain, and they can also be used in gluconeogenesis to maintain blood sugar. The other thing to note is that all of these reactions are reversible. That means that these processes can also be utilized in the synthesis of particular amino acids from the corresponding keto acid. So not only can you break down amino acids to glucose, but you can also take glucose and other intermediates and form amino acids. Now, the degradation of protein produces bicarbonate, and it produces ammonium ion. The bicarbonate comes from the complete oxidation of the carbon skeleton, which produces carbon dioxide in the TCA cycle. The ammonium ion comes from the deamination of amino acids, as we showed just a minute ago. Now, this becomes a problem. Let's say that you ate a 16-ounce steak. That's a 1-pound steak. Hydrolysis of the proteins yields a mixture of about 725 to 730 millimoles of amino acids. If you catabolize all of these amino acids, you'll produce about 2.5 moles of carbon dioxide, about 1 mole each of ammonium ion and bicarbonate, and about 25 millimoles of sulfate. This would increase the concentration of ammonium and bicarbonate to toxic levels in your blood by about 20 millimolar. The ammonium ion is toxic, and that much bicarbonate would raise your blood pH to greater than 7.6. So the body has to deal with this excess ammonium and carbon dioxide somehow. And the way that it does that is through a process that we call the urea cycle. This is a series of chemical reactions which primarily take place in the liver. And what this does is it converts 1 molecule of bicarb and 2 molecules of ammonium ion to urea, which is a much more soluble, a less toxic molecule, which is then safely excreted by the kidneys. The cycle also produces a proton that's able to titrate another molecule of bicarb. So in the process, we remove 2 molecules of ammonium ion and 2 molecules of bicarbonate. This is the urea cycle, and again, it's a complicated cycle. I don't need you to remember all of the steps, but just a couple of them, which are outlined here: the carbamoyl phosphate synthetase and ornithine carbamoyl transferase. The rate-limiting regulated step is the synthesis of carbamoyl phosphate from bicarb, ammonium ion, and 2 ATPs. The enzyme responsible for this is carbamoyl phosphate synthetase 1, or CPS1. This step is regulated by a compound called N-acetylglutamate. N-acetylglutamate synthesis is increased when we have a high-protein diet. So this N-acetylglutamate that stimulates the urea cycle to deal with this greater protein load. It does it because it's an allosteric activator of the carbamoyl phosphate synthetase 1. Now, the urea cycle can have inherited defects, one of which is illustrated in the following clinical vignette. A newborn baby boy is brought to the nursery after birth. The nurses note that he vomits after every feed, and the vomiting becomes gradually intractable and is finally accompanied by convulsions. He begins to feed poorly and becomes very lethargic. Physical exam is significant for hyperreflexia, but is otherwise normal. The neurologic exam is significant, again, for hyperreflexia, and physical exam is otherwise normal. The labs show normal CBC, normal chemistries, normal LFTs, but his ammonia level is very high. You're suspecting a particular genetic defect, and so you do special urinalysis exam for orotic acid, which you detect in the urine. This patient has a disease called ornithine transcarbamylase deficiency, or OTC deficiency. OTC deficiency is an X-linked recessive disorder, so mostly seen in boys, and is caused by a deficiency in the second step of the urea cycle, which is ornithine transcarbamylase, or another word for it is ornithine carbamoyl transferase. This is the most common of a group of diseases known as the urea cycle defects. The most damaging consequence of these defects is the accumulation of ammonia, leading to ammonia intoxication. And this causes symptoms including vomiting, convulsions, lethargy, and poor feeding. Left untreated, these patients will progress to coma and death. Now, these diseases are variable. The age of onset and the disease course depends upon the severity of the defect. If it's a complete enzyme deficiency, you'll get problems immediately after the first protein load in the first postnatal feed. These infants have extremely high mortality. The less severe of defects may cause symptoms only during a period of illness or only with consumption of a higher protein diet. And so patients with less severe defects can be treated with low protein diets. Now, treatment may include any of the following measures. You may give them a low protein diet. You can give levulose or lactulose, which acidifies the colon and prevents the absorption of ammonia. That reduces the ammonia load. You can give antibiotics that kill ammonia-producing bacteria in the GI tract, again, the purpose of which is to decrease the ammonia load. You can also give dietary supplementation with arginine or citrulline, which increases residual urea cycle activity. Summary of the urea cycle: the inputs are bicarbonate and ammonia. The output is urea. The key enzyme is carbamoyl phosphate synthetase 1, which is regulated by N-acetylglutamate, which is increased in high protein loads. Associated diseases include urea cycle defects, the most common of which is ornithine transcarbamylase deficiency. This is the end of the section on general amino acids.